Last time we left off, we were just finishing up talking about respiration, and we talked about the electron transport that's associated with mitochondria. And there's a couple of things that I wanted to point out that are different between what happens in plant mitochondria and what happens in animal mitochondria. There are two main differences. All animal mitochondria have the NADH dehydrogenase complex, complex 1, which takes electrons away from NADH and puts them into the electron transport chain. And in association with that, pumps protons across the membrane. Plants have, I think there are at least 3 known now, alternative NADH dehydrogenases, which put electrons into the same electron transport chain, but do not pump any protons across the membrane. That seems somewhat wasteful, doesn't it? Because the proton gradient that's being produced is leading to the production of ATP, ultimately. So having NADH dehydrogenase enzymes that don't pump protons would seem to be wasteful. But they're there, and plants that don't have those don't do as well. They don't die, but they don't do as well as plants that do have them. The other end of the electron transport chain, normally the electrons go to oxygen through complex 4, the cytochrome oxidase complex. Plants have a complex referred to as the alternative oxidase, or AOX. The alternative oxidase complex is basically doing the same. It's taking electrons away from the electron transport chain, but it's doing it much earlier, and there's no proton pumping associated with that either. Okay, so plants have 2 different mechanisms, one for getting electrons into the electron transport chain, the other for getting electrons out of the electron transport chain to oxygen, that avoid proton pumping. And the book talks about the conditions under which this should be favorable for the plant. And I want to emphasize that you think about that and make sure it makes sense to you. Because on the surface, it would seem that this would be something that would be wasteful. Taking the same energy input from NADH and getting substantially less ATP out of it. Why, under what conditions might that be beneficial for the plant? Okay, so the alternative electron transport chains are one of the interesting things that distinguishes respiration, mitochondrial respiration in plants from that in animals. We talked about the regulation that occurs, the overall regulation of carbon flow through glycolysis in the citric acid cycle that's largely controlled by ADP. When ADP accumulates, it goes up and activates enzymes early in glycolysis to put more carbon through there to make more ATP. So when ADP accumulates, we decided this accumulates under conditions where the demand for energy is high. ATP is being used up, ADP is accumulating. That turns on enzymes at the beginning of glycolysis to promote more production of ATP. So remember, the key thing that you should take away from this diagram is that energy supply in any organism, this is not unique to plants, energy supply is determined by demand for energy. If the cell doesn't need energy, it slows down respiration. If it needs more energy, it speeds up respiration. It's not the other way around. Timmy?
Wait, are you going to explain why the lack of proton pumping associated?
Am I going to explain that to you? No. I want you to look at it. I want you to think about it. It might be fodder for an exam question. Okay? You have to do some thinking on your own as well. Okay. So the other aspect of regulation of photosynthesis, or regulation of respiration that we talked about that is unique to plants has to do with this region between hexose phosphates and triose phosphates. Because in non-photosynthetic tissues or in non-photosynthetic organisms, there's no sucrose synthesis. There's no big pools of triose phosphate coming out of the chloroplast that are going, under most cases, mostly towards sucrose synthesis. That doesn't happen in animals, right? So it shouldn't be surprising that the regulation of processes between the hexose phosphate and triose phosphate pools will be different in plants than it is in animals. Because animals never shuttle carbon in this direction. In animals, it's always going in that direction towards glycolysis, or at least almost all the time. Okay, so why has this become important? Well, there are enzymes that are present in plants that are not present in animals. The interconversion of triose phosphates and hexose phosphates is typically accomplished by these two enzymes, which either takes off a phosphate or adds on a phosphate, depending upon which way you're going. Plants have a completely different enzyme that pushes things in the direction of glycolysis. And it shouldn't be surprising to you that there's a unique type of regulation here. And I think we mentioned this briefly at the end of lecture yesterday, that the regulation is accomplished by an interesting sugar, fructose 2,6-bisphosphate. The intermediate that functions between triose phosphates and hexose phosphates is fructose 1,6-bisphosphate. So this is an intermediate in carbon metabolism. This is a regulatory molecule. And this regulatory molecule, the amount of it determines whether carbon is going to flow towards sucrose or whether carbon is going to flow towards glycolysis. So this is a level of regulation that never happens in animals because there's no big flow of sugar out of the chloroplast into the triose phosphate pool towards sucrose synthesis. That just doesn't happen in animals. Yeah?
Don't animals have gluconeogenesis generating glucose?
Yep.
So, but they're regulated also by fructose 2,6?
Nope. Nope. No, gluconeogenesis occurs by a slightly different pathway, and it's not regulated in the same way. Right? And in addition, certainly you're not in animals. You're not making sucrose. You're making it most hexose phosphates. Okay, so there are several differences in how respiration functions between plants and animals because of this interplay between photosynthesis and respiration. And you should at least have a feeling for why these different types of regulation are important. You should be able to explain it to me. Okay, let's just finish up then by asking a simple question, at least on the surface, a simple question. We're going to make a graph of the change in oxygen concentration as a function of time. So change in oxygen is a function of time on the y-axis. And on the x-axis, we're going to put light intensity. When we start in the dark, there's no photosynthesis going on, but there's respiration going on. So we would expect, if this is 0 here, there's going to be a net uptake of oxygen, and this represents the dark respiration rate. And as we increase light intensity, we're going to see something that looks like that. And we said that this point where it crosses 0 is referred to as the compensation point. It's where photosynthesis equals respiration. They just happen to balance each other. Okay, so nothing in this diagram should be surprising to you. The question is, if we make a plot of the respiration rate as a function of light intensity, what does that look like? Is the dark respiration rate, is that constant for all light intensities? Does the graph look like this? Or does something else happen? If you look in the literature, particularly in ecological literature, there's the assumption that the dark respiration rate is constant as a function of light intensity. But all the measurements that have been done to look at this give a very different picture. The actual rates of dark respiration as a function of light intensity starts off at at relative, here's the dark respiration rate, is light intensity increases the respiration rate in the mitochondria. So we're measuring now separately of what's happening in photosynthesis and respiration. This mitochondrial respiration actually decreases, stays relatively low, and then about where photosynthesis starts to get saturated, the mitochondrial respiration rate starts to go up. So at high light intensities, the actual mitochondrial respiration rate is substantially larger than it is in the dark. Why might that be the case? How could we rationalize that respiration rates, cellular respiration rates, mitochondrial oxygen uptake might be higher at highest light intensities than it is in the dark?
Is there a saturation before class?
Say that again. One more time.
Like saturation of the chloroplast.
I'm sorry.
Like saturation of chloroplast.
So there's, sure, the chloroplasts are saturated, but why does that, what does that have to do with mitochondrial respiration rate?
Well, they won't produce as much ATP.
What won't produce as much ATP? The chloroplast, the mitochondria? I'm not following what you're talking about. No?
Well, it means that we have higher respiration when we go down.
That's right. So this is
we
higher
produce, we produce more ATP. Now.
We need more, yeah,
So that could be because we maybe need to produce more proteins for like heat protection. And stuff.
Yeah, so protective processes to deal with reactive oxygen species and stuff like that, that's certainly a part of this, yes. What else?
To use up some of the products of
Use up products of photosynthesis, and what at high photosynthetic rates, what else are the products of photosynthesis being used for besides sucrose? Glycolysis, but why? Right? Yes, so this is higher rates of cellular respiration. That includes glycolysis, but why? What else is going on there? What else could be going on there? What else does respiration do for the cell besides provide energy? Carbon skeletons, yeah. All the other metabolism at high light intensities, that other metabolism is cranking along faster to try to use up the products of photosynthesis to build more cellular stuff. Okay? So the supposition, the assumption that mitochondrial respiration is constant in light is incorrect. Whether or not this makes huge differences, well, depends. At lower light intensities, probably not too big a deal, but at higher light intensities, it certainly is important. Yes?
So why does it go down first before it goes up?
Why does it go down? Good question. Why? How can you rationalize that it goes down? What does this mean in terms of the cellular respiration's production of energy for the cell under these conditions? It's decreased, right? How could that make sense? Why might that make sense?
Because you're producing fewer sugars and equal running photosynthesis.
Producing fewer sugars, so you mean sucrose, or do you mean?
Well, I mean, you have fewer products of photosynthesis, so there's going to be less respiration to break heat out.
But there's no products of photosynthesis here, right?
Oh,
Right? So the question is, as we're actually making more products of photosynthesis, as light intensity goes up, why does respiration rate go down? What you suggested seems to be backwards to me.
Is there more energy being used for photosynthesis, and that's why respiration will go down?
See if you can carry that a little bit further. Because remember, the energy that's being used for photosynthesis is being produced in the chloroplast and used in the chloroplast.
Right.
But does it stay in the chloroplast? Do the products of photosynthesis stay in the chloroplast? No, they're exported. right? So as light intensity increases at the low levels, you're having, let's go back to this diagram here, as light intensity increases, you're getting inputs into glycolysis, intermediates in glycolysis that weren't there before. In the dark, this isn't happening. And as you start to turn the light on, this starts to happen. I'm seeing a lot of confused faces. Think about this. See if it makes sense to you. Again, it would be a good question to put on an exam to rationalize why this decrease in respiration rate happens. Remember, the whole idea of this class is to get you to think about this stuff.
But if you don't explain it in 2,000,
Yeah, okay, so there's two ways we can do things here. I can explain to you every single thing that I want you to know. Or I can challenge you to think about some of the things on your own, right? Did I explain to you every single thing that I asked you on the last exam? No, right? So I'm not going to do it, not going to start now, right? I want you to be able to think about some of these things. Okay, so questions other than explaining why the respiration rate goes down as light intensity increases. Any other questions about respiration? Yes?
I'm just a little bit confused about that chart.
Yep.
So you were saying that when it's below the zero axis, there isn't any photosynthesis happening.
This is in the dark. Remember this, okay, I understand where there might be some confusion here. This graph here is the net change in oxygen. And it represents, we're just putting an electrode in and measuring how much oxygen is either taken up or produced. That's the sum of what's happening in photosynthesis and respiration. Using stable isotopes, it's easy to separate out oxygen production in photosynthesis versus oxygen uptake in respiration. So this is a graph of the actual oxygen uptake associated with respiration. Dark respiration right here, that oxygen uptake decreases but doesn't go to zero at low light intensities. And as we get to saturation, that oxygen uptake actually increases again. So this is looking at just mitochondrial uptake of oxygen. This is the net of photosynthesis plus respiration. So if you want to know what the curve for photosynthesis looks like, you take the difference between these two.
So
Wait, sorry, go ahead.
is
Grace.
that one graph, the top graph, is that the amount of oxygen that's being produced?
That's the net change in oxygen. So at this point right here, it's zero. But does that mean there's no oxygen being produced? No. There's oxygen being produced by photosynthesis, but it's being taken up at an equal rate by respiration.
So it's basically the net oxygen produced, so below it is, you have negative.
This is net oxygen uptake.
Net oxygen
Yep.
uptake.
Right? So remember when we looked at, for example, when we compared C3 and C4 plants, we saw that the C4 plants had a compensation point that was much lower than C3 plants. Right? That's because they're more effective taking out CO2, doing more photosynthesis at low light intensities and low concentrations of CO2.
So since that graph is increasing, you can assume that there is more photosynthesis as the...
Oh, sure. There's definitely, as you turn up the light, photosynthesis increases. Absolutely. But it's not until this intensity that you see net oxygen evolution. Below that, respiration exceeds oxygen production. And so the net is negative. You okay with that? Francesca, you had a question.
Yeah, I was just wondering if this is more a time thing that the curve goes up and down below zero, or if that stays actually that low respiration rate, if the light stays that low.
It doesn't go to zero. So first of all, respiration never produces oxygen.
No, but I mean, it goes it's fairly low at that point,
Yep.
right? So is that only light dependent, or is that also time dependent? So if we have a long time where the light's like this, will it stay that way?
Oh,
or-
that's a good question. Yeah, that's
it kind of looks
yeah.
like it could be something to do with, getting things started or something.
Yeah, that's a really good question because these sorts of measurements are made in relatively short-term exposures. So basically you take your plant in the dark and you measure the oxygen exchange, and then you expose it to a low light intensity, and you wait 5 to 10 minutes, let things come to steady state, and you measure the things again. But if you kept it at that low intensity for several hours, yeah, the respiration rate could change because maybe it's not meeting the metabolic demands of the cell over time. Yeah. So this is like a snapshot at relatively short periods of time at these different light intensities. That's a good point.
I just want to make sure I understand the axes. So there's still photosynthesis occurring even when it's dark, when light intensity is below 0?
Is there, when the light intensity is below 0? No,
So
no.
which axis is what, I guess?
This axis is change in oxygen as a function of time. This axis is light intensity. This is 0 light intensity. So at 0 light intensity, that's the dark respiration rate.
Oh, okay. So
As
it
you
goes up.
increase the light intensity, photosynthesis goes up, but it's not until you get to the compensation intensity that there's more oxygen evolution from photosynthesis than there is oxygen
So
uptake from
the
respiration.
x-axis is not really the light intensity, right?
This is light intensity, yes. That x-axis is light intensity.
Right. Okay. But you still
Okay.
have light before you cross it. That makes sense now.
Yeah, so there's oxygen evolution at this intensity right here. It's just the amount of oxygen evolution is less than the oxygen uptake in respiration. So the net is negative. Okay? That makes sense?
Yeah.
All right. Let's move on then to talk about nitrogen assimilation. Okay. So we know that nitrogen is important for plants. When we talked about mineral nutrition, we talked about nitrogen as being a very important source mineral nutrient for plants, one that's often limiting in real environments. So this is why farmers tend to put a lot of high nitrogen fertilizers on their soils, because the amount of nitrogen that's normally available in the environment, even in a natural forest, nitrogen is often limiting to the ability of plants to grow. So it's worthwhile us thinking about how do plants get nitrogen and what do they have to do with it in order to be able to make use of it to make proteins and amino acids and things like that. So let's think first about the various forms of nitrogen that are available in the environment. The most common type of nitrogen is nitrogen gas. And that's N2. And if we think about the valence of nitrogen in these different compounds, the valence of nitrogen gas is 0. The most common form of nitrogen that's available, nitrogen gas is not available to plants. Plants cannot make use of this as a nitrogen source, even though there's tons of it out there. The most common form of available nitrogen is nitrate ion. That's NO3 minus. And its valence is, what is it? It is +5. The valence of nitrogen in organic compounds, like amino acids, this would be something like this, where R is some organic group, and the valence here is -3. There's another -3 valence that we're familiar with, ammonia. And there's one more positive valence form of nitrogen, nitrite ion. Okay, so nitrate is the form in which nitrogen, most plants obtain nitrogen from the soil, and we'll see why in just a minute. But the reason I'm talking about these different valences is for you to recognize that if the plants are taking nitrate up from the soil, if this is the form of nitrogen they're getting, before they can incorporate it into organic compounds, they have to reduce the nitrate to the level of ammonia. So there is an 8-electron reduction involved in converting nitrate to ammonia before it can be incorporated into organic acids. 8 electrons, that's equivalent to 4 NADHs or 4 NADPHs. Energetically, that's a lot of energy. Right? So plants, this is going to be a significant investment on the part of the plants to be able to not just find the nitrogen in the soil, but to take that nitrogen they get in the form of nitrate and reduce it to ammonia before it's incorporated. Okay, so one of the things that you learn about in an ecology class or an introductory biology class is the idea that these different pools of nitrogen are not constant. The nitrogen just doesn't sit there all the time. There's a lot of fluxes between various pools. So if we think about plants, they're taking up nitrogen from the soil. But animals eat the plants. Some of the plants die and the tissues decay. The animals create waste products that contain nitrogen in it. The animals die. So there's nitrogen that is cycling around this all the time. And we need to understand this cycling in order to understand what might limit the availability of these different products for plants to take up. So this looks relatively complicated. I have a much easier global nitrogen cycle here where we've got atmospheric nitrogen up at the top. And remember, I said there's tons of that compared to these other forms of nitrogen. Ammonia, nitrite, and nitrate are all available in the soil, although the vast majority of it is in the form of nitrate. And I just gave you the answer why in a very indirect sense. Why is nitrate as opposed to ammonia? Well, let me point out that when organic material dies and decays, the nitrogens that are in amino acids, when those amino acids decay, are released into the soil as ammonia. So you might think, okay, there'd be lots of ammonia in the soil. There is not. Why not? Yeah, it gets oxidized. And why would the bacteria oxidize it? For energy, right? So look at, we just said that plants, to convert nitrate into ammonia, have to put 8 electrons in, right? That costs energy. What happens if we run this reaction in the opposite direction? Get that energy back, right? So there are bacteria in the soil. They're called nitrifying bacteria. And they're sitting out there waiting for the ammonia to come along. And as soon as it comes along, they convert it to nitrate and use the energy derived from that to run their metabolism. They're chemoautotrophs. They're not getting their energy from sunlight. That would be a photoautotroph. They're getting their energy from reduced compounds in the environment, in this case, ammonia. So the reason that nitrate is what's most common is because ammonia is, let's say, unstable. Because of biological organisms that basically use this as free fuel, that ammonia is rapidly converted into nitrate. So which one would be energetically more favorable for plants to take up? Nitrate or ammonia? Ammonia, obviously. It's just that there isn't much ammonia around under most circumstances. That's why they would prefer to take up nitrate. Okay? So understanding, in particular, this component of the nitrogen cycle is important because it tells us that even though decay of organic material produces ammonia, ammonia is typically not available for plants in the soil. There's some there, but not much. If you're working at a farm and you're fertilizing the soil, very often the fertilizer you put in contains some ammonia and some nitrate. In fact, it's very common to use ammonium nitrate as fertilizer. Anybody else know what else you use ammonium nitrate for? Say that again? Ice packs? I don't know. Are those ammonium nitrate? They could be. I don't know. The most common place they're used is in explosives, like depth charges. How does an explosion happen underwater if there's no gaseous oxygen there for it? It's because ammonia and nitrate, this has got an oxidant, this has got a reductant. You put enough energy in to get it started, you got an explosion there with electrons moving back and forth between them. Yeah, so that's why fertilizers are commonly used by terrorists for bombs, because it's a cheap way to make a pretty easy explosive. Okay, one other thing we need to think about as far as this picture is concerned, and that has to do with the properties of ammonia. Ammonia, first of all, let's recognize that ammonia gas can react with water to form ammonium ion plus hydroxyl ion. Right? And so when we talk about ammonia or ammonium, we should think about them as being equivalent in terms of nitrogen content anyway. They're obviously different in terms of charge and pH and things like that, but they're basically equivalent with each other. But ammonia is toxic for living cells. And the reason it's toxic at micromolar concentrations is because it breaks down pH gradients. pH gradients like those that are important in ATP synthesis. So ammonia is a small molecule. It's very permeable through membranes. You don't need any transport proteins to get ammonia gas across there. But if you have ammonium ion in a high pH region, it tends to protonate, forming ammonia gas. And that ammonia gas can easily diffuse across the membrane into a low pH region where it picks up a proton. Right? So we started off with ammonium on this side. Now we got ammonium on this side. It was being driven basically by the concentration gradient. But the net reaction is uptake of an OH over here and uptake of a proton over here. It's decreased the gradient across the membrane, the proton gradient across the membrane. And that's why ammonia is dangerous. It basically breaks down proton gradients that are needed for chemiosmotic ATP synthesis. So one of the things that cells have to do is very carefully regulate how much ammonia they produce. As soon as they produce ammonia, they got to incorporate it into amino acids, into organic compounds. Otherwise, it can accumulate to toxic levels. Okay, so an important question. Before all the reactions associated with reducing nitrate to ammonia and stuff like that can happen, you got to get these nitrogen-containing compounds into cells. Right? You got to get it across the plasma membrane, get it into the roots so it can be translocated to all the other cells. How does it get into the roots? How would you expect nitrate to get into the roots? Remember back when we talked about ion movements and the fact that calcium and magnesium were at lower concentrations in the cells than we expected? How about nitrate and phosphate? What were the concentrations of those inside the cells compared to Nernst equilibrium? Remember the answer to that? There's a lot more inside the cell than you'd expect if it was at equilibrium. 10 to 100 times more nitrate inside the cell than what you'd expect based on the redox or the membrane potential across the plasma membrane. So if there's more inside the cell than you expect at equilibrium, what's happening? Active transport. The cells are expending energy to get nitrate into the cell. And that's normally what happens. There are other mechanisms that can get nitrate in when the nitrate concentration in the soil is very high, non-physiologically high, farmers' fields sort of high. So there's non-active components that you can have, but they usually function only at unusually high nitrate levels. For most cells, The normal levels of nitrate that are present outside require active transport to get it into the cell. Okay. How about ammonia? Any ammonia transport proteins? Probably not, yeah, because ammonia is a small membrane permeable molecule. It can go through the membrane just the way water can, right? Doesn't need a channel, doesn't need any transport protein. So for ammonia, there are no dedicated transport proteins for ammonia, but for nitrate there are.
You mentioned that ammonia is toxic to cells.
Yep.
So why would they kind of allow toxic substances, or do they convert it to something as soon as it enters?
Yeah, right. So we're going to see that they reduce nitrate to ammonia, but that ammonia never accumulates. It's immediately taken up and incorporated into organic compounds. But that balance, obviously, has got to be important. If that balance gets screwed up, the cell can poison itself with too much ammonia. Okay? Okay, so let's think about the processes that are going on inside the cell to take nitrate ion and reduce it to ammonia. That's the first step. Once the nitrate's inside the cell, that's the first step in getting that nitrogen into a form that can be used for organic compounds. And the reduction of nitrate to ammonia is a two-step process. It's catalyzed by two different enzymes. The first one is nitrate reductase. Often abbreviated NR, and it catalyzes the reaction so it's taking nitrate and reductant, and that reductant is either NADH or NADPH, and converting that reversibly to nitrate ion plus water plus NAD or NADP. Two electron reduction from valence of +5 to valence of +3 is catalyzed by this nitrate reductase enzyme. Couple important things about nitrate reductase. Its activity almost always limits how much nitrogen is available to the plant. That is, if you give the plant plenty of nitrogen, the thing that limits how much nitrogen can be taken up is nitrate reductase activity. Okay? So if you want to measure how much nitrate the plant is taking up, measuring nitrate reductase activity should be a good indicator of that. So when how do you increase the nitrate reductase? We'll talk about how it's regulated in just a second. Right? So what you'll see very often when you look at physiological studies of nitrogen assimilation in plants is people will measure the amount of nitrate reductase activity because it is a direct measure of how much nitrogen becomes available to amino acids and nucleic acids and those sorts of things. Okay, so how is its activity regulated? It's a pretty interesting enzyme because it's activated by nitrate. As surprising as it may seem, relatively few enzymes are activated by their substrates. That is, that there's a regulatory site that the substrate binds at that's different than the active site that turns on the enzyme. The other thing that you might expect is that nitrate reductase is inhibited by ammonia, very effectively inhibited by ammonia or ammonium ion. Why does that make sense? Why does it make sense that nitrate reductase is inhibited by ammonia? It's the end product. Yeah, there's one. That's part of it, but it's not the whole story.
Sorry, I just had a question. Doesn't it, the reaction that gets rid of the NO2 minus reduce ammonia?
It does. Yeah, it does. But remember, I said ammonium and ammonia are rapidly interconverted. So let's just treat them as the same thing. What's the other thing? So Patrick correctly pointed out that ammonia is the end product of this process. And the accumulation of that end product would feedback inhibit everything else that's going on. But there's something else that's more important.
It's toxic.
No, I'm not sure that the fact that it's toxic would explain why it inhibits.
Is it like, does that have to do with energy?
Yeah, it has everything to do with energy. It takes 8 electrons to reduce nitrate to ammonia. If ammonia is around, do you want to be reducing nitrate? No. Why invest the energy if ammonia is around? So, I mean, obviously we're giving a teleological explanation for something that evolution is selected for. But it's not surprising that evolution selected for ammonia inhibition of nitrate reductase if it saves the plant a lot of energy. Don't reduce nitrate if you don't need to. If there's plenty of ammonia present, you don't need to reduce nitrate. The enzyme is also highly regulated by phosphorylation. So phosphorylation, sticking a phosphate on, phosphorylation inactivates and removing the phosphate activates it. And the conditions that phosphorylate and dephosphorylate it are largely related to light. And why that makes sense, why you would want to link this to light isn't apparent at this point, but it will be in just a minute. I should point out that this enzyme is cytoplasmic. It's located in the cytoplasm in all types of organisms that do nitrate reduction. It's a cytoplasmic enzyme. So there's this phosphorylation, the activation and inactivation is largely regulated by light, and we'll see why that's the case in just a minute. Nitrate reductase has one other very bizarre characteristic, and that is the enzyme has a very fast turnover time. That is, if we measure how long from when a nitrate reductase enzyme is synthesized, when it comes off the ribosomes to be a functional nitrate reductase, how long before that nitrate reductase protein is broken down, the average lifetime of a nitrate reductase enzyme is 1 to 3 hours. They don't hang around very long. And so that's a bit of a problem, because that means the cell is investing a lot of energy to make nitrate reductase. It costs a lot of energy to make proteins, and it costs energy to break them down. Why is it doing that? And it should be clear that there has to be some cost to the cell for having too much or not enough nitrate reductase around that makes it beneficial to have it controlled that quickly. What that cost is to the cell, we don't really know. But the fact that it has a very short turnover time, it only exists for 1 to 3 hours, tells you that the amount of enzyme that's present in the cell at any given time is something that's important for the cell. It needs to be able to ramp up and ramp down the amount of nitrate reductase that's present in the cell very quickly. One of the things that you're probably starting to see, and I can guarantee you by the end of the semester you're going to be sick of, is that we're going to be talking about things that we know less and less about. Right? So we don't know why nitrate reductase is this. Wait till we talk about signal transduction pathways and plant hormones. We know what the hormones do, sort of, but when we talk about the signal transduction pathways, the way I like to describe them are 1,000-piece jigsaw puzzles where we only have 10 pieces, and we're trying to figure out what the picture is. Right? This is going to become more and more common, so get used to it. I'm not going to be able to explain everything to you because plant physiologists don't understand how a lot of these things work in detail. It's just the way it is. We're trying to invite all of you to become PhD students in plant physiology and figure out the answers to all these problems. Okay. So let's look at the next step of this process, and that is reduction of nitrite to ammonia. And that's catalyzed by the enzyme nitrite reductase. That's abbreviated NIR. And nitrite reductase is a plastid enzyme. If nitrite is reduced to ammonia in the leaves, it happens in chloroplasts. If it's reduced in the roots, it happens in proplastids, the progenitors of chloroplasts, because there's no chloroplasts in the roots. Nitrite reductase, the reaction that it catalyzes. So we take nitrite. And what else do we need here? We need 6 reduced ferredoxins. So remember, ferredoxin is an electron carrier that functions after photosystem 1. And we also need a bunch of protons. And this is going to give us ammonia plus some waters plus some oxidized. So if this isn't 8, this should be 6. Sorry. 6 oxidized ferredoxins. So this is a fairly expensive enzyme for the cell to use. And the fact that it is a plastid enzyme makes a lot of sense if the nitrite is reduced to ammonia in the leaves, because the ferredoxin is produced as a product of photosynthetic electron transport. Right? So when this reaction happens in the leaves, the electrons coming out of photosystem 1, there's a competition between those electrons being used to reduce CO2 and make sugars, or to reduce nitrite to ammonia to make amino acids and things like that. So an interesting experiment you can do, it's easier to do with algae than it is to do with higher plants. Take a culture of algae that's nitrogen starved. Measure the photosynthesis rate that's going on for example, is CO2 fixation. And then add nitrate to the culture. When you add the nitrate to the culture, the rate of CO2 fixation drops quickly. And the reason it drops is because if the cells are starved for nitrogen, when you give it nitrate, the cells have to reduce that nitrate to nitrite and then nitrite to ammonia. And the reduction of nitrite to ammonia uses electrons that come out of photosystem 1 that would otherwise be used to reduce CO2. Okay, so it's a very simple experiment to illustrate the competition between carbon reduction, CO2 reduction in photosynthesis, and nitrite reduction associated with nitrogen assimilation. That same competition doesn't happen directly if this reaction happens in the roots of plants. If it happens in proplastids in the roots, those roots aren't doing any photosynthesis. The ferredoxin is produced by starch metabolism in the proplastids in the roots. So there's no direct competition with photosynthesis, although it still requires energy. The energy is just being taken from starch rather than directly from photosynthesis. Okay. So the energy cost, there's no way to get around that. In leaves, that energy cost is met by competition with CO2 fixation. In the roots, it's met by metabolism of starch in the plastids. And now we can see why it makes sense, at least in some circumstances, that nitrate reductase is regulated by light. That in the light, the nitrate reductase enzyme, the phosphate is taken off of it, and it's activated. In the dark, the phosphate is stuck on, and it's turned off. Because, at least in those plants where nitrite is reduced in the leaves, there's a big light requirement here. Right? don't reduce nitrate to nitrite unless you got an electron source to reduce the nitrite to ammonia. If the nitrite is being reduced to ammonia in the roots, the light regulation of nitrate reductase doesn't make it anywhere near as much sense, and yet it is still there. Another question for plant physiologists.
Fairly low light conditions for whatever reason.
Yep.
It's going to be cloudy for the next 10 days.
Yep. You mean like Ithaca?
But you never trust the weather service here,
Yeah.
so you keep them.
Okay.
If I spray nitrogen on the leaves, like nitrite directly
Yep.
on the leaves, to serve to have more photosynthesis that can actually reduce carbon instead of
Yep.
doing nitrogen, would that help me?
Would that help your plant under low light conditions? Yes, because what the plant will do, and this is a really interesting to study. One of you wrote, one of the questions you wrote me this morning was about partitioning between CO2 fixation and nitrogen assimilation. And even in low light, it will partition some fraction of the electrons. If you look at, for example, in an algal cell, algae don't make all the cellulose stuff that plants do, so it's easier to study there. If you look at an algal cell, the ratio of carbon to nitrogen in an algal cell is typically about 16:1, right? So that tells you that if the cells have to have competition between CO2 fixation and nitrogen assimilation, for every 16 electrons that go to fix CO2, there's going to be you need 3 times as many here. So there's going to be a significant amount of electron flow that's going to nitrogen assimilation even under limiting light conditions. If you took that nitrogen away, the rate of photosynthesis would actually go up. The rate of CO2 fixation would actually go up in the algae because you've removed the competition for reduction of nitrate. Okay, so we have now got the nitrogen that came into the plant as nitrate reduced to ammonia. But we said ammonia is toxic to cells. It can't hang around. That ammonia has to be incorporated into organic compounds fairly quickly. And the question is, how does that happen? And the reactions that accomplish that, the main reactions in plants that accomplish this, it's different in algae, but in plants, the main reaction is a pair of reactions. A reaction that starts with glutamate. This is an amino acid. There's the amine group up here. And it sticks this ammonium on as a second amine, converting glutamate to glutamine. And that reaction requires ATP, energy associated for ATP. So now we've taken a compound with one NH2 group and made it into a compound with 2 NH2 groups. So that's the first step. That's the step in which the nitrogen is actually incorporated into an organic compound. But the next step is equally important because now we take one molecule of glutamine in one molecule of oxyketoglutarate. You may not remember, but this was an intermediate in the citric acid cycle. And we transfer this amino group to this one, and we end up with 2 glutamates. So the net of this reaction is 1 ammonia plus 1 oxyketoglutarate makes 1 glutamate, and the other glutamate just cycles around. So the utilization of these 5 carbon compounds from the citric acid cycle is the net input of carbon into making a whole suite of amino acids. The book talks about another reaction, a reaction that actually is much more important in most algae, glutamate dehydrogenase. And I think 1 or 2 of you asked, the book talks about the glutamate dehydrogenase usually runs in the other direction. It's instead of putting an ammonia on, it takes an ammonia off. Why would you ever take an ammonia off of an amino acid? Why would a plant ever do that? Let's make it more general. Why would you ever do that? Why would you ever take an amino acid and take the ammonia off of it?
This is why we say nitrogen is mobile and it's taken all of these?
That's a possibility, yeah. It turns out, though, when we talk about mobile nitrogen, it's usually not moved around as ammonia or nitrate. It's moved around as amino acids. But that's a perfectly legitimate suggestion.
Maybe you're eating this?
Yeah, basically you're eating it and you're converting the carbon skeletons into things that are going into respiration. Yeah, that's basically what glutamate dehydrogenase is important for. So if you starve yourself, the first thing you do is you use up all the sugars in your blood. Then you use up all the starch stored in your muscles and your liver. And once that's gone, the only carbon skeletons you have to live off is to break down proteins. And to get proteins in, you've got to break them down from glutamate back to this compound. And that's what glutamate dehydrogenase is. So that's the reason that enzyme is there in most organisms, is to run this backwards, in case you have to take carbon from amino acids and get it back into glycolysis in the citric acid cycle. But this, for plants, is the main mechanism by which ammonia is assimilated. And notice there's an energy requirement as far as ATP here, and there's an energy requirement for reductant here. So there's a big cost, not just with the reduction of nitrate to ammonia, but the incorporation of ammonia into organic compounds. There's a real cost to the cells for this. So it's not surprising that these processes are carefully regulated. Plant isn't going to waste energy if it doesn't need these nitrogen compounds. Okay. Let's see. We got enough time. Let's skip this. And I want to talk about nitrogen fixation. So back in the beginning, when we talked about the main pool of nitrogen that's available on Earth is atmospheric nitrogen, but that plants can't make use of it. But there are some organisms that can. Those organisms are only prokaryotes. There are no eukaryotes that can use atmospheric nitrogen. There's several different types of bacteria, some of them photosynthetic bacteria, some non-photosynthetic, that can do this process called biological nitrogen fixation, that convert atmospheric nitrogen into ammonia. There's also industrial processes. So one of the first Nobel Prizes went to a guy named Haber, a German, who figured out how to use energy from burning fossil fuels primarily to convert atmospheric nitrogen into ammonia, how to make fertilizer, basically. Lightning can do it. So just lightning in the atmosphere can convert some of the nitrogen gas in the atmosphere to nitrate. Okay, but we want to focus on this, and you know that this is important. You know that from an ecological perspective, the process of nitrogen fixation is important because it's bringing unavailable nitrogen into a pool of available nitrogen. So, for example, if you're a farmer, it's a lot less expensive to allow living organisms to do this for you rather than buying fertilizer. So green fertilizers, organisms that can do this nitrogen fixation is really important. So I just want to spend the rest of the time talking about this overall process of biological nitrogen fixation. There are two basic types of situations where nitrogen fixation occurs. And one very interesting one is in cyanobacteria, blue-green algae. These are prokaryotes, photosynthetic prokaryotes, that do oxygenic photosynthesis just like plants do. So they have basically all the same electron transport stuff, all the same CO2 fixation. But in addition to that, under nitrogen-limiting conditions, certain cells in this branch, this filament of single-celled algae, can differentiate to form what's called a heterocyst. And a couple of you said in your questions this morning, wait a minute, I didn't think that bacteria could do this. It didn't make multicellular organisms with different functions. Well, some bacteria can, and it shouldn't be surprising to you because bacteria are the progenitors of multicellular organisms. So it shouldn't be surprising to you that some bacteria can do things, differentiation into different cell types, that are more like what happens in multicellular organisms. All the rest of these cells are referred to as vegetative cells. They're capable of doing photosynthesis and all the normal things. The heterocysts are very much specialized for nitrogen fixation. Okay. A different type of bacteria, non-photosynthetic bacteria, can do nitrogen fixation in a symbiotic relationship with plant roots. Not all plants. Legumes are the common ones, beans and peas and things like that. And we'll talk about the formation of these root nodules and what's going on in that symbiosis in just a minute. But the bacteria that are in these nodules and the cyanobacteria are basically accomplishing the same reaction. The enzyme that catalyzes this is an enzyme called nitrogenase. A very interesting enzyme. The reaction that it catalyzes, let me get this right, it takes nitrogen gas and it takes 8 electrons and it takes 16 ATP and it takes 8 protons and it makes 2 ammonia plus hydrogen gas plus ADP and phosphate, and I think that's it. So 16 ATP and 8 electrons. Very expensive process. Why is it expensive? Because remember, nitrogen gas has a triple bond in it. And breaking that triple bond is hard to do. So this enzyme catalyzes this reaction. So what would you think about, let's not think about the blue-green algae, but let's think about the bacteria that are doing this in plant roots. What would you think about the amount of respiration that is going on in those bacterial cells that are carrying out this nitrogenase reaction? Lots of respiration, right? Lots of oxygen needed to support that respiration. Make sense? Well, the bizarre thing is that nitrogenase is irreversibly inhibited by oxygen. So that presents a bit of a dichotomy. You have an enzyme that's inhibited by oxygen, but the same cells that nitrogenase is functioning in need a lot of respiration in order to produce the energy that nitrogenase needs to run. How do we account for this? It's all happening in the same cell. It's a prokaryotic cell, so there's no mitochondria or separate organelles you can do these things in. Once you understand it, it's relatively straightforward, but it's something that plant physiologists stumble over all the time. It's the problem of, we talked about the bucket problems, water flowing into and out of the bucket. So let's have our bucket be the concentration of oxygen in the cell, right? And we need lots of oxygen for respiration. Right? So there's going to be a lot of oxygen flowing out of this bucket, right? So if we don't put any oxygen in, the bucket's going to get empty really quickly, right? Right. But if we put oxygen in by diffusion, such that the rate at which oxygen is coming in is just equal to the need, then you can have the bucket being pretty empty. You don't have to fill this bucket up. As long as the rate of diffusion matches but is not greater than the rate of respiratory uptake, the cell doesn't need to have much oxygen in it. And this is how it works. That's the basic mechanism. There are some interesting things that happens in these cells. If you read the textbook, you heard about leg hemoglobin. It's an oxygen-binding protein. Also tends to keep the oxygen concentration down. But the basic mechanism is, as long as the rate of diffusion does not exceed the rate of oxygen uptake, this bucket will not fill up. It's only if it's diffusing faster than it's being used that the bucket will fill up. So it's actually not that complicated to have cells that must have high respiration rates, big demand for oxygen, and at the same time keep the oxygen concentration low. Okay, the last thing I want to talk about is in many ways not really related to plant physiology at all. But it's a really interesting phenomenon, and that is to think about the symbiosis that's going on between these soil bacteria that are capable of nitrogen fixation in the plant. It's not hard to understand what that symbiosis involves. The nitrogen-fixing bacteria are giving reduced nitrogen to the plant, and the plant's basically giving fuel, sugars, to the bacteria. So there's exchange of materials between the two things. But the one thing that you want to keep in mind in this type of symbiosis is the same that we talked about with mycorrhizae. There are lots of bacteria in the soil who would love to get all the carbon they can from the plant without giving the plant anything. We call those bacteria pathogens. So the question is, how does the plant distinguish symbiotic bacteria from pathogenic bacteria? In fact, many very closely related bacteria to the rhizobia that are involved in nitrogen fixation are pathogenic. So the question is, how does this relationship, this symbiotic relationship, become established in a way that guarantees to the plant that it's getting the right bacteria, it's allowing the right bacteria to interact with it? And we're going to talk just very briefly sort of the upper level of this. There are two courses taught here at Cornell that cover nothing but the communication between host and symbiont when symbiotic relationships form. It's very complicated, not surprisingly, because the host organisms have to protect themselves. Okay, so one thing that's important to keep in mind is that this tissue, the nodule, is plant tissue. They're cells that were in the roots that for some reason, from signals that are going on there, started dividing again and producing these new cells that leave these root nodules. The bacteria that are doing the nitrogen fixation are inside those cells. They're not actually in the cells, but they're in vacuoles in the cytoplasm. So there's some interesting stuff going on here that I want to try and briefly talk about. So anybody read Mad Magazine? Nobody's even, you guys are all too young. Well, you've heard of it at least, right? When I was your age, Mad Magazine was all the rage. And there was a cartoon in there called Spy versus Spy. And it's talked about all the things that one guy did to counteract what the other guy did, to counteract what the other guy did, just all this interesting communication. This is the same sort of thing. There's a lot of communication that's going on to make sure that the bacteria that are going to end up inside some of these cells are the right bacteria. Okay, so that communication starts off by the plant roots. These are free-living soil bacteria. So how do they know what the right plants are? Because not all plants can form these symbioses. Well, we'll see that in a couple lectures that plants produce a lot of compounds that are involved in limiting competition. Plant roots release compounds into the soil in order to keep other plant roots from growing too close. Some of those compounds that are produced are in the category of flavonoids. We'll talk about these in a couple lectures. The flavonoids that are released by legume roots are signals to the rhizobia. There's a legume nearby, come this way. So there's a chemotactic movement of the rhizobia from the soil toward the plant in response to these compounds that the plant is releasing, not to signal the bacteria, but to keep other plant roots from growing too nearby. Okay, so the rhizobia come near the plant, and when they get near the plant, when they detect the presence of the plant, they produce compounds. These compounds are called nod factors, and here's the they're pretty bizarre structure. There's a fatty acid stuck on this. There's a nitrogen-containing sugar. There's a bunch of these nitrogen-containing sugars, and so it's a weird structure. It has nothing to do with anything that's normally present in the plant. The nod factor is a signal from the bacteria to the plant, basically saying, "Get ready, we're coming." Right? Get ready for all the things that you need to do to go from taking a normal plant root to making these root nodules. There's a lot of cell division that's got to go on, for example. Okay, so what do the nod factors do? The first thing the nod factors do is cause the root hairs to grow out, and they literally curl around and sort of trap the rhizobia in the soil. The other thing that happens is right in this region where the root hair curls, the cell wall of the root hair breaks down, exposes the plasma membrane. And the next thing that happens is really bizarre. The plasma membrane at this point where it's exposed folds in. So basically, you have the plasma membrane like this with the bacteria out here, and then the plasma membrane does this. It folds into the cell and allows the bacteria to move into this, what's called an infection thread. This whole thing here is an infolding of the plasma membrane. The one thing that you want to keep in mind throughout this process is that the bacteria are never actually in the cytoplasm of the cell. Notice, if this is an infolding of the plasma membrane, these bacteria are still actually outside the cell. If these membranes broke off and made little vesicles containing the bacteria, they'd still technically be outside the cytoplasm. This should look pretty familiar to you. This is the exact same sort of process that gave rise in an evolutionary sense to chloroplasts and mitochondria, right? Okay, so there shouldn't be anything too surprising that stuff like this can happen. The one thing that's really bizarre is this infection thread grows all the way through the root hair cell, gets to this boundary between the root hair cell and the next cell inside there, and the plasma membrane fuses, releases the cells outside the cell, and the plasma membrane invaginates on the next cell. So basically, they're released outside the cell here, and then the next plasma membrane invaginates, and the process continues into several other cells. So this thread grows from one cell to the next, providing a mechanism for the bacteria to get from the outside to the inside of the root without actually getting inside a cell. Now, something that's not shown in this picture, but is a consequence of the nod factor, the nod factor is another thing it signals to have happen, is for these cells in the interior of the root, they start dividing. Those cells will be the new home for these bacteria. Those cells are the cells that give rise to these nodular structures. The cells in those nodules are filled with little vesicles. Whoops, sorry, going the wrong way here. They're filled with little vesicles that contain one or more of these bacteria. And within these vesicles is where the symbiosis is happening. The plant is giving sugars by transporters that are present in the vesicle to the bacteria, and the plant is getting from the bacteria, again via transporters that are present in that membrane, nitrogen-containing compounds. So one of the things I'd like you to think about, one of the things that I could put on an exam, would be to tell me about what's happening. This membrane that surrounds the bacteria is derived from the plasma membrane of that cell. It folded in and pinched off. What must happen in that membrane in order to allow this symbiosis to occur? There's got to be a lot of changes that happen there. And you should be able to give me an idea of what some of those are. But we're just really touching the surface of the communication that's going on between the bacteria and the plant to establish this symbiosis. There are something on the order of 60 different proteins that mutations in those, the genes that encode those proteins, screw up this symbiosis. There's a lot of chemical communication going on to make sure that this happens correctly. And it shouldn't be surprising that that's the case. Okay, we'll stop there and we'll pick up with the next topic, which I think is plant secondary products, making compounds like flavonoids by the roots. We'll do that next Tuesday.